So this is video 1.1D, and we're going to be on page 9 and 10 of your biology handbook. We're going to take 1 minute to review what we talked about last time. Last time we started talking about 2 of the 4 macromolecules that all living things need. Now remember that macro, M-A-C-R-O, macro, means big. So 4 big molecules, or fairly big, at least compared to a cell, molecules that we need to live, to survive. The 2 macromolecules that we talked about yesterday, or last time, was carbs and lipids. It's really important for us to understand the monomer of both of those, to know and to be able to name and identify the monomer of carbohydrates and of lipids. We also need to know the elements that make them up. And we also really need to know the function and indicator test. And the indicator test that detects whether or not the carbohydrates are there or if the lipids are there. Now, as a quick review for carbohydrates, what was the name of that monomer? Well, that was your monosaccharide. For lipids, you had your triglycerides. So in this case, you just really need to remember this. Create yourself a chart, create a study guide to be able to remember those monomers. One thing, or carbs and lipids had several things in common. One of them were the elements were the same. CH and O, both of them had the elements CHO. And for function, both of them can be used for energy. So that was also another thing that they had in common. The difference in the functions, however, is that for carbohydrates, that's short-term energy, and they can be used, carbs can be used for structure within a cell, like the cellulose of a plant's cell wall. On the other hand, for lipids, their energy use is for long term. So if you think about a bear that eats a lot before it hibernates, well, that's long-term storage of that energy. The other thing that lipids can be used for is insulation. So thinking of that same bear, the more fat it stores, the more insulation, the more warmth it has. And then for our indicator test, this was also different. For carbohydrates, we had 2 tests to be able to determine if small carbohydrates were there or if large carbohydrates were there. We can use Benedict's for small carbs like glucose. We can use iodine for large carbs like starch. Whereas for lipids, we had a very simple test. It wasn't even a chemical. It was actually the brown paper bag. Wetting the brown paper bag with your sample, if you're not sure if there's a lipid on it, holding it up to the light, and if it's a little clear, kind of transparent, then that's going to be showing you that there are lipids in your unknown sample. Same thing with the carbs. If your sample is unknown, what you're testing for, you don't know what's in it, and you want to see if there's small carbs or large carbs, you can use Benedict's or iodine. But for today, we're actually going to start talking about our last 2 macromolecules. The first one is a nucleic acid. We saw this word in our vocab sheets. Nucleic acids are atoms, or nucleic acids are molecules that are made up of the atoms CHNOP. Okay? C-H-N-O-P, CHNOP. That's your carbohydrate, your carbon, your hydrogen, your nitrogen, oxygen, and phosphorus. So this one's different from our carbs and lipids. We don't just have the elements CHO. Okay, we don't have just the 3, but we actually have 5 of those 6 essential elements. They're arranged into and that says 3-part monomer. Let me see if I can move this over for you. They're arranged in a 3-part monomer called a nucleotide. So here we have these 5 atoms. They bond together, and they're going to form a structure, and this says 3, that's made of 3 parts. And that monomer is called a nucleotide. Just like how in carbohydrates we had the monomer monosaccharide. And in lipids we had the monomer triglyceride. Well, nucleotides come in multiple different types and can store information in their sequence. And we'll talk more about this sequence later. But right now, we're mainly concerned about the elements that it's made up of, the monomer that it's made up of, and of course its function. For DNA, for nucleic acids, there's actually two types. So right here I'm going to write two types, and I want you to do the same. There's two types of nucleic acids. The first is deoxyribonucleic acid, and you know that as DNA. DNA is a double strand, two strands of nucleotides twisted together. And those nucleotides, those monomers, are going to carry the code for cells to make their needed molecules-like functions. So carrying the code for cells, that's going to be our function. And that's actually going to help us, or code for proteins. Code for proteins. And I just drew a little arrow right there to help us memorize that. In our notes, you have a couple of pictures. And in one of those pictures, you have this structure down here. And it's inside of a box. And you can actually see that it's labeled as a nucleotide already. Go ahead and take a highlighter or a colored pen or a crayon or a colored pencil. And please put a box. Go back over that box that says nucleotide. This is the name of the monomer of nucleic acids, and you need to know the three parts of that. So I'm just going to label it in these different sections. The P right here stands for phosphate. That's part one. The second part here is going to be number 2, our sugar. And the third part here, and notice this third part looks a little different. There's different letters here. There's A's, T's, C's, G's. And this is going to be our nitrogen, and this is going to be number 3, nitrogen base. Okay. Those three things, our phosphate, our sugar, our nitrogen base, those are going to make up our nucleotide, which is the monomer of our nucleic acid. The second type of nucleic acid, and this is a new slide right here, so make sure you're paying attention to this. This is number 2, second type of nucleic acid, and this is called ribonucleic acid. Ribonucleic acid stands for RNA. It's actually going to be a single strand of nucleotides. So instead of having these two strands, and by the way, on this picture, let's label this. On our picture that we started labeling, let's go ahead and label that as DNA, because that's not RNA. DNA equals double strand. RNA is going to be single strand. So RNA will actually only have half of this picture, of this figure. So RNA is a single strand of nucleic acids that performs different jobs to help DNA make proteins. So help DNA make proteins. This is the function. DNA has the code to make the proteins, the instructions, and RNA is going to physically help make those proteins. One thing we do need to know is there's going to be no indicator test for nucleic acids. Okay, no indicator tests for nucleic acids. There are tests that can determine whether or not DNA is there or detect DNA, but not a test that we'll be able to use in class. And so the tests that we're talking about, the indicators we're talking about in class, we can actually perform in class. For DNA, it's more technical, and we just don't have those resources at school. But our actually the last of our 4 macromolecules is going to be proteins. And proteins are atoms of CHNOPS, all 6 proteins, Proteins are atoms of synapse arranged into a monomer called an amino acid. So for proteins, they're made up of all 6 of our elements, of our essential elements, synapse, all 6. And the monomer for our proteins are known as amino acids. Amino acids come in 20 different types. You don't need to know the names of the 20 different types. You just need to know that there's lots of them. And they must go into the right order to form the right shaped protein. For proteins, shape is very, very important. In this picture, what they're showing you is the different names of those amino acids. Usually it's represented as some sort of chain, or you can think of a string of beads as representing the protein. And here they're showing you that they actually fold into a specific shape before they become a protein, or a usable protein. So a protein shape is important to the job it performs, and there are 6 important jobs. Here, here we want to underline that shape is important. This is important to know. And here are our different functions. Okay. Proteins can be used for structure, the shape of something. It can also be like your hair, your nails, your muscles. It can be used for communication, like with hormones like insulin that help regulate your blood or your blood sugar. It can be used for immune protection, prevent you from getting sick. It can be used to absorb light, like the melanin in your skin, or the pigments that are in your eyes, or chlorophyll that are in plants. It can help with transport, like the hemoglobin in your blood that transports oxygen around your body. It can help. It can perform as an enzyme, like catalysts that breaks down hydrogen peroxide, enzymes that speed up chemical reactions, which we'll talk more about next time in our lecture. But one other big thing that you need to know about proteins, and I want you to write this really big, is that proteins do not provide energy. Lots of times we get this mixed up. They do not provide energy. People that take a lot of protein or drink or eat a lot of protein before they work out, well, they're trying to build more muscle. The protein itself is not going to provide you with energy, and that's one thing that would really get mixed up, but that's something you have to remember. Okay? The only 2 molecules that provide us with energy would be what? What were the first 2 that we talked about? Carbohydrates and lipids, and that's it. Okay? So make sure you write that really big somewhere in your notes, at the top of the page if you need, or on the side if you have a little extra room. And the last thing that we need to know about proteins is our indicator test. So this is our test again. It's going to indicate if protein is present. The name of this indicator test is body rent. It starts off as a blue color and turns to a purple color. These 4 macromolecules are found in every living thing on Earth. Cells make and break these down as part of the cell's regulation and homeostasis needed for survival. One thing here, homeostasis, I want you to define it, write the definition as the constant internal conditions to be in balance, and we'll put in parentheses, or normal. Okay? To try to maintain homeostasis means that the body, the organism's body, whether that's an ant or a human, is trying to regulate what's normal for it. So if our body's supposed to be at a certain temperature, we're going to try to stay at that certain temperature. If we're supposed to have a certain amount of water in our bodies, then we're going to try to maintain that certain amount of water. The last thing that I want you to do is to fill out your summary box, and I want you to write 2 facts about macromolecules. So that can be about the carbohydrates, that can be about the nucleic acids or the proteins, because that's the two that we want to focus on today. And that'll actually be all for your lecture today. Make sure you go and check your knowledge.